Greetings, everyone, um, members of AFRH, both home and abroad. On behalf of the Central Executive Committee of the AFRH, we welcome you to this webinar series, which has been on for some time. I'm Dr. John Ekwani, a consultant gynecologist, and myself and Dr. Femi Akisoya will be co-leading this session in the first series of genetics in this webinar series of the AFRH. We thank all of you for making yourself available to make our time to make these events possible in this period. We appreciate everyone, including the Central Executive and the Secretariat, for making this event possible. So we thank all of you for coming. I don't know whether Dr. Akisoya can just say hi so that we can hear his voice and know that he's with us.
Good evening, everyone. I'm Dr. Femi Akisoya, and I'm highly privileged to be part of this panelists. And I want to say that we're in for an exciting moment and a wonderful topic to be discussed. Thank you for joining. Thank you for coming. You're welcome.
All right. Thank you so much, Dr. Akisoya. We appreciate. We're going to be considering a very important topic that is quite crucial in the management of infertility in our environment. The genetics of male and female infertility and management, and it's going to be ably presented by Mr. Uwa Joseph Olayinka, who I'll be reading his profile shortly. But before then, I just want to lay some ground rules that will be quite beneficial while we present. I will implore all of us that have joined this webinar series to please mute our mics. Make sure that there is no interference from your background that will make the platform very noisy and making it difficult for us to listen to the presenter. So I will enjoin all of us to please mute our mic, be attentive. And then we have a chat option at the bottom of your screen, which enables you to make some comments and especially post your questions, which will be ably read out and addressed by my colleague, Dr. Femi Akisoya, who will be handling the question and answer session at the end of this presentation. And I believe if we do this, we're going to have a very neat and tidy presentation at the end of the day. So once again, thank you so much for coming along. I'll be reading the profile of our presenter, who is Mr. Yinka Uwa. Joseph Olayinka Uwa is a distinguished senior embryologist and geneticist with over 10 years experience specializing in pre-implantation genetic testing and transport IVF, and has made significant contributions to the field of reproductive medicine. He serves as an international editor, board member for innovation in scientific tech and social ecosystems, and holds a postgraduate diploma in assisted reproductive technology from the University of Schleswig-Holstein, Kiel, Germany. Olayinka also holds a master's degree in genetics and leadership in health management. He is the head laboratory services of Fertigold Fertility Clinic, and alongside provide professional support to other fertility institutions. He is a member of a number of professional bodies, such as AFRH, ACE, ASRM, ESHRE. He has, with his skills and dedication, contributed in improving the chances of success for countless couples facing fertility challenges. His passion for educating and empowering others in the field of reproductive medicine is evident through his dedication to knowledge and dissemination. Ladies and gentlemen, it gives me good pleasure to welcome Mr. Joseph Olayinka Uwa to take the floor. I think you will have about 20 minutes time for presentation, if I'm not mistaken. And at the end of it, we'll be taking your questions and answers for you to address. So you have the floor, Mr. Olayinka. Thank you so much.
Thank you very much, Dr. John Ekwani. Thank you very much, Dr. Femi Akinsoya. It is indeed a great pleasure for me to be here to talk about this very important topic, genetics of male and female infertility and management. I hope we can hear me well before I commence.
Yes, please.
Dr. John, can you hear me?
Yes, I can.
Okay, thank
You're
you very
loud
much.
and clear.
So, okay, I also want to thank the AFRH for giving me this platform to speak on this subject already expressed by Dr. John Ekwani. And the subject is genetics of male and female infertility. And these are my outline. So first, infertility is relatively a common health condition, and it affects nearly 7% of all couples worldwide. Heterogeneous pathology with a complex etiology that includes both environmental and genetic factors. And primarily, it has been estimated that about 30% of infertility cases are due to genetic defects or genetic factors. And several studies have shown with animal knockout models convincingly that infertility to be caused by gene defects, both single gene and multiple gene. However, despite enormous efforts, progress in translating basic research findings, there are still some gray areas in the research or study of genetic infertility, or what genetics contribute to both male and female infertility. And the genetic causes remain unexplained for the vast majority of male and female infertility patterns. And a particular difficulty is the huge number of candidate genes to be studied. From the genome project, there are still a lot of genes that needs to be studied, and these genes, they influence reproductive function in humans and could contribute to a very good aspect of male and female infertility. And at present, there are only a handful of genes or genetic defects that have been shown to cause or to be strongly associated with primary infertility. Yet with completion of the Human Genome Project and progress in personalized medicine, the situation is rapidly changing. And indeed, there are new genes being discovered on average and being added to clinical genetic testing lists annually. So there are still some challenges. However, it's still a study that is going on and a lot of research. So the objective really is from the World Health Organization. The report states that about 15 to 17.5% of adult population in the world is infertile, and genetic abnormalities are thought to account for about 30%, like I said, of male and female factor infertility. So it is very important for us to identify underlying genetic basis. One, to establish the cause and diagnosis. Secondly, to personalize therapy. Thirdly, develop appropriate screening tests that can be used to counsel couples planning to utilize assisted reproductive technology. So, genetics of infertility, we cannot talk about male and female infertility without actually looking at the origin. So, there are different aspects of the genetics of infertility, from the one that is most bogus, which is the chromosome, to the genes, which are nucleotides that get expressed in real world. And the nucleotides are multiple segments of DNA-based pairs. So what the Genome Project has done is to analyze all the human base pairs. I think about 300 billion base pairs. And this has been a research that started from 2000 and it finished about 2013, and it consists of several countries: the United States, UK, Germany, Italy, Japan, and China. And they've been able to actually look at these DNA-based pairs. But the challenges are still enormous, because we are not just looking at the chromosomal contribution of infertility, but you are also trying to look at the epigenetics and also look at the gene contribution, and that is where we are right now. So we'll be looking at genetic male infertility. Genetic male factor infertility is a situation where men are infertile as a result of gene defects or chromosomal abnormalities. And like I said earlier, for genetic, there are contributing factors. One, you look at the chromosomal disorders. Two, you look at mitochondrial DNA mutations. Three, you look at monogenic disorders. And we also may be considering endocrine disorders of genetic origin. And first, there are some lists. Like I said, we'll move from the chromosome level to the chromosomal aberrations that contribute to genetic male factor infertility. And we'll be considering just a few, which are the common ones. We have Klinefelter syndrome. Then we'll be looking at maybe Y chromosome microdeletion. There are still some others as a result of translocation, testicular disorder of sex development, and complex structural chromosomal aberrations. All these are the common chromosomal abnormalities that contribute to genetic male infertility. So let's look at Klinefelter syndrome. Actually, this Klinefelter syndrome is one of the most common aneuploidy in males, and it was first described in 1942. It is characterized by a list of at least one superior X chromosome, resulting in the karyotype 47XXY. So if we look at the regular pairs of chromosomes, the 23 pairs of chromosomes, we have two sex chromosomes, the X and the Y. However, in the case of an individual that presents as Klinefelter, there's an extra X chromosome, and this extra X chromosome actually affects the man that presents this Klinefelter syndrome. And This syndrome is characterized by the presence of, like I said, one or more extra chromosomes. And the karyotype 47XXY is the most prevalent type. The prototypic man with Klinefelter syndrome has traditionally been described as, they are usually tall. I know some of us must have seen these kind of men in our society, in our environment. They are tall with narrow shoulders, broad hips, sparse body hair, and gynecomastia. If you look at their waistline to their belly, it's almost like shaped like that of a woman. And for private assessment, they have small testicles, androgen deficiency, and they usually present as azoospermic men, and decreased verbal intelligence. And not until 1996, men with Klinefelter syndrome were considered infertile. But with the development of TESE, which is testicular sperm extraction, and the intervention of ICSI, it has now been possible to extract viable spermatozoa from the testes by surgical biopsy, and injection is done. The sperms are injected into oocytes, mature oocytes. And worldwide, more than 60 children have been born after successful ICSI in couples where the male partner has Klinefelter syndrome. And then Klinefelter syndrome is, in clinical practice, is, we can usually come across them, and they is a very common genetic disorder that contributes to genetic male infertility. We'll go to the next, which is this is most common in our society, where we see people that present to be azoospermic, and about 10 to 20% of males without sperm in their ejaculate carry a deletion of the Y chromosome. It's known as the Y chromosome micro deletion. And these deleted regions include the Y chromosome azoospermia factor, that's the AZF. So if we can see my pointer, it's showing a different region. So there are basically three regions. We have the AZFA, we have the AZFB, and we have the AZFC. And is located in the long arm of the Y chromosome, the 11 region, and which is divided into three, like I said. And the gene that provides instruction from this, when there is a mutation in any of this region, either the mutation in the A region, the B region, or the C region, it affects man. And research has shown that is a very is a common factor that contributes to genetic male infertility. And the missing genetic material likely prevents production of a number of proteins that is needed for normal sperm cell development. And so these men, they present as azoospermic men. Like I said, it plays a major role in genetic male infertility. And the expression, or the physical expression, is azoospermia. So I would probably, when we want to ask questions, we'll spend more time in explaining the contribution of this particular genetic disorder, and whether it can be transferred, because it's a Y chromosome microdeletion. So that means it has to do with male. And so when a client goes through IVF treatment and is azoospermic and eventually is being treated, is there a chance that that gene can also be transferred to the children, to the offsprings, the male offsprings? We are going to hopefully answer that when we are dealing with questions and answer. Another genetic factor under single gene disorders. Now, like I said, we have the chromosomal disorders, which has to do with the 23 pairs of chromosomes. So you can have mutation or an aberration or deletion, addition in any of those chromosomes, and it can affect. What we just spoke about is a mutation in one of the sex chromosomes, which is the Y chromosome. But now we want to go into the single gene disorders. So these are some of the single gene disorders. We have the congenital bilateral aplasia of the vas deferens, known as the cystic fibrosis, and we have mitochondrial DNA mutation in human sperm. We have androgen receptors mutation in androgen receptors. We have mutation in the aurora kinase C. We have mutation in HSF2, which is the heat shock transcription factor 2. But the research in these other ones are not so much, because, like I said, not so much is known about the contribution of gene or gene mutation in genetic male infertility. And so many research are still ongoing. But let's look at the role of Klinefelter in genetic male infertility. So we have, I beg your pardon, cystic fibrosis in genetic male infertility. So like I said, cystic fibrosis is caused by a mutation in the gene, which is the CFTR gene. Healthy men with cystic fibrosis, so men usually present themselves and they may look healthy. So the most common mutation is a deletion of the 3 nucleotides that result in a loss of the amino acid phenylalanine. And cystic fibrosis is the most common fatal genetic disease affecting in Europe. It's not so common amongst Negroids, but among Caucasians is a very common genetic disease in men. And healthy men with cystic fibrosis gene mutation may have problems of the vas deferens development, and that is a congenital absence of the vas deferens, or they may have blockage of the tube with mucus. You can see from the let me just move this. So we can see from this diagram, the mutant CFTR channel does not move chloride ions, causing sticky mucus to build up on the outside of the cell. So you have the inner cell and the outside cell. So the presence of the blockage or the mucus prevents flow of chloride ions. Whereas if you look at the normal CFTR channel move chloride ion to and fro. And many of us must have heard about obstructive and non-obstructive azoospermia. Cystic fibrosis play a major role in this. And healthy men with cystic fibrosis gene mutation may have problems of vas deferens development, and the blockage of this tube with mucus. Affected men are usually present as azoospermic, and like I said, mostly obstructive azoospermia, because sperm cells are made but are not released during ejaculation, and this is as a result of the blockage. And secretion of electrolytes and water by epididymal epithelium is important in the formation and optimal fluid environment for sperm maturation and transportation. From the point where sperms are being stored to meet the seminal fluid, and they get transported after being released. This process is disrupted in the genetic disease cystic fibrosis caused by mutation of the CFTR gene, like I explained. So is a major cause of obstructive azoospermia. And lastly, for genetic male infertility, looking at what role mitochondrial DNA mutation contributes. Mitochondrial DNA mutation in human sperm is the 16569 BP double-stranded circular DNA molecule. That's the base pair. And the spermatozoa need a great deal of energy. So when sperms want to move or when they are swimming, they require energy. And that energy, is generated by the presence of the mitochondria, which is the ATP that is required for sperm movement. And this is found in the midpiece region of the sperm. So spermatozoa, they need this energy to support their rapid movement after ejaculation. And random attacks on naked mitochondrial DNA of sperms by reactive oxygen species or free radicals will inevitably cause oxidative damage or mutation to the mitochondrial genome with pathological consequences. And this lead to infertility in males, because where sperms are unable to swim to meet the egg as a result of poor sperm motility, then we will now be seeing cases of clients presenting to be infertile, whereas it's a mutation that has occurred in the mitochondrial DNA. And oxidative damage or the mutation of the mitochondrial genome cause pathological consequences and lead to infertility in male. The midpiece of the mammalian sperm contains 70 to 80 mitochondria, and there is one copy of mitochondrial DNA in each mitochondrion. So about 85% of sperm sample contain large-scale mitochondrial DNA deletions of variable sizes, and most spermatozoa have 2 to 7 deletions of mitochondrial DNA. So mitochondrial DNA plays a very major role, or mutation of the mitochondrial DNA plays a very major role in sperm motility. And where there is a deletion in mitochondrial DNA, it has been pointed out that an age-related increase in oxidative stress and oxidative damage to mitochondrial DNA is a major cause of male infertility. So mitochondrial DNA plays a major role in the motility of the sperm. And like we know, atenosospermia, it prevents the sperm, or the sperm is unable to penetrate through the mucous filled cervix to reach the site for fertilization. And this is usually caused by the absence or aberrations in mitochondrial DNA, because once there's a mutation in the mitochondrial DNA, then ATP that will help the sperm generate energy for the sperm cells to swim will not be able to, the sperm will not be able to perform that function optimally. So we'll be moving now to genetic female infertility. Like I said, it's the contribution of male and female infertility is 50-50%, but not so much is known about the genetic effect of female infertility compared to male infertility. However, we are going to look at some chromosomal defects or chromosomal mutations. We are going to also look at some gene mutations that contributes to genetic female factor infertility. So genetic female factor infertility is a situation where women are infertile, and it can be as a result of chromosomal abnormalities or gene defects, like I said. So chromosomal aberrations, single gene disorders, polygenic disorders, and we'll also be looking at some endocrine disorders of genetic origin. So let's look at some chromosomal aberrations in genetic female infertility. One, you have the most common, which is trisomy X. That's the 47XXX. Another is Turner syndrome. That's monosomy X. Then we have the CNVS, which is a genomic aberration copy number variant. Then we'll also be looking at X chromosome rearrangement. We'll be looking at S chromosome mosaicism if time is on our side. So let's look at trisomy X, which is the 47XXX syndrome. This is just like a sister of the Klinefelter syndrome, which is the XXY, as a result having an extra Y, extra X chromosome. For trisomy X, you have three, instead of two X chromosome, you have one extra X chromosome, which makes it trisomy, and is one of the most common causes of premature ovarian insufficiency. It occurs in about 1 in 1,000 female births, and the majority of women with trisomy X, they present to be normal. It's the aberration happens due to chromosome non-dysfunction errors in meiosis 1 or meiosis 2 during oogenesis. So we can see that there is, during meiosis 2 stage, or sometimes it happens in meiosis 1, there is non-disjunction, and instead of the diploid cell, diploid X chromosome to divide into 2, there is a part where, as a result of the non-disjunction, the X chromosome does not divide, and at that point you have another X chromosome joining the S chromosome. So instead of having one pair of X chromosome, you have one extra X chromosome. So like I said, the aberration happens as a result of chromosomal non-disjunction error, and it results in triple X syndrome. These females, they present to be normal, and the only common genetic defect, or the only expression, is a few of them suffer from premature ovarian insufficiency and malformation of the genitourinary tract. Next, we'll look at the one that is most common, and that is Turner syndrome. Is the contrary of trisomy X, which is monosomy X. It has a lot of variation. The Turner syndrome is a genetic condition. It affects females and is strongly associated with infertility due to ovarian dysgenesis and premature ovarian failure. It presents at puberty and menstrual defects, and sometimes early menopause. this is a case where instead of having the 2X chromosome, like we can see the 46 pairs of chromosome with the XX chromosome, which in Turner syndrome, it presents in different ways. It can present as a monosomy, that is 45 chromosomes with one X chromosome. It can also present in a mosaic form. The mosaic form can either be 45X or 46XX. And you also have another mosaic form where it presents as 45XX with one loss of chromosome. And lastly, it can also present as 45X or 46XY. So this condition, usually it affects the women a lot, and it affects a lot of the reproductive process in women. And most women with Turner syndrome, like I said, they have ovarian dysgenesis, and therefore they are usually infertile. And in very rare cases, you find spontaneous menses followed by early menopause. And only 2% of the women have natural pregnancies, with high rates of miscarriages, stillbirths, and malformed babies. So Turner syndrome play a major role in genetic female infertility. And next, we'll be looking at some single gene disorders. We have the Fragile X syndrome, galactosemia, primary ovarian failure, which is POF, and so on and so forth. There are lots, but like I said earlier, the gene disorders, the research is still ongoing to know particular genes, because you have multiple genes sometimes contributing to affect or cause mutation, contribute to genetic female factor infertility. So lots of research are still going on to know particular genes that are involved in a particular genetic disease. But let's look at one that is common, which is the Fragile X syndrome. It's caused by mutation in a single gene, the Fragile X mental retardation, that's the FMRO1 gene, and is located on the X chromosome. Is a disorder characterized mainly by mental retardation, and also in the Fragile X mutation, it results in, like I said, the Fragile X mental retardation gene that is switched off, and at that point in time is the FMRP, which is the Fragile X mutation result, is no longer produced, and it prevents normal neurodevelopment. Some of the symptoms of Fragile X syndrome are severe, but it suffices to say it also expresses itself in male. In fact, in male, in where it happens in males, it's usually severe, because males only have one X chromosome, whereas for female they have two X chromosomes, so one can complement for the other. So because females have two chromosomes, and an inherited damage of the FMRO1 gene can be compensated for by an inherited LDFMRO gene, that's from the other X chromosome. So unlike females, males only have one X chromosome, like I said. So more, the condition is more severe in males. And this protein is required for brain to develop normally, and it plays a role in the development of synapses, specialized connections between our nerve cells, which are critical for relaying nerve impulses. And approximately 20% of females with this mutation, they develop premature ovarian insufficiency, that's POI, which can also be called premature menopause. And about a third of females with this mutation, with this defect, experience cessation of menses on or before the age of 29, and about 1% will have it at age 18. That's those that present with Fragile X syndrome, severe Fragile X syndrome. So it is recommended that female carriers of this syndrome are alerted to the risks of what they will be exposed to. And really, currently there is no cure or no treatment for Fragile X syndrome. Only recommendation are behavioral therapy. Another is primary ovarian failure, which is defined as either complete or incomplete failure of the ovaries. It's also known as POI. That's primary ovarian insufficiency, like I stated earlier. And a number of genes have been associated with the primary ovarian failure. X-linked genes include like I said in the previous slide, the FMR1 and the bone morphogenetic protein 15. These are genes that have been associated with primary ovarian failure. However, these genes also, they don't only play a role, if you look at this diagram, they don't only play a role in primary ovarian failure, they also have other, they have also been seen in other abnormalities. And so chromosomal gene abnormalities have long been regarded as cause of POF, but the percentage vary widely among reported series because, like I said, the role of the gene itself compared to if there is a chromosomal mutation, genes they have contributing factors, and you have several genes that come together to cause a particular genetic defect. So genetic heterogeneity and disease development have been widely investigated and observed to have close relationship. However, with the development of sequencing technology and increasing solid evidence suggests that genomic instability underlies pathogenesis of primary ovarian failure. So if it's clear, we understand that there are a lot of genes involved in primary ovarian failure, and also some of these genes don't only affect primary ovarian failure or insufficiency, they also play a role in other diseases or defects. So it's very hard to know the particular gene that is involved in this. So it's females with these defects usually present themselves, and they have poor they have failures in their ovaries and poor ovarian reserve. Then like I said, we also look at some polygenic complex female infertility. The roles that how it affects endometriosis, how it affects polycystic ovarian syndrome, and some receptors that are involved in both LH and FH, and FSH, that's mutation in LH and FSH receptors. All this, they contribute to, they have contributing factors, polygenic factors that affect female infertility. And so the genetic association and linkage studies have identified some candidate genes, and follow-up studies are needed to replicate the studies and narrow down critical regions, because these regions have been found not to only affect endometriosis and polycystic ovarian syndrome, they also affect other regions, or cause other diseases. Like, so polycystic ovarian syndrome, trying to move my, is a complex endocrine disorder with, like I said, heterogeneous genetic causes. So studies of multiple candidate genes can see some of the genes: FB, N3, SFT, INS, PCOS1, and so on and so forth. They have been seen or associated with polycystic ovarian syndrome. However, these genes, they've also been shown to have significant association with other disorders. So research are still going on to know the particular set of genes that actually affect the or cause endometriosis, or cause polycystic ovarian syndrome. Nonetheless, there are genes, there are genetic defects, mutations in genes that have been associated with these disorders. So before we end, we'll talk about some diagnosis techniques and management options. We'll look at 3 stages: karyotype analysis. We'll also look at fluorescent in situ hybridization of spermatozoa. Then we'll look at PGTA, PGTM, and PGTSR. The karyotype analysis refers to the set of chromosomes found within each cell in human species, and the karyotype consists of 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes that define the sex of the individual. Like I said, we have 23 pairs of chromosomes, and we have the sex chromosome. For male, you have the X and Y. For female, you have the XX. So therefore, karyotype analysis is used to study whether a person has the chromosomal load that corresponds to him or her. On the other hand, it presents some type of translocation, trisomy, monosomy, etc. So if there is any mutation in the chromosome, with karyotyping, that can be detected. So the karyotype is performed on both members of couples and has an approximate cost, and more and more fertility clinics are indicating the performance of a karyotype at the first visit to make a more personalized diagnosis. So when a client presents and there's suspicion of a genetic disorder, with karyotype analysis for both the man and the woman, you can tell whether there is any mutation in the chromosome. Fluorescent in situ hybridization, which is FISH of the spermatozoa, is another technique used to analyze possible abnormalities in the number or the structure of the chromosome. Like I said, you can do that by cryotyping. You can also do that using fish. And as it can be performed on spermatozoa, the diagnostic, but this can be performed on the sperm. And then this diagnostic test provides more information about male reproductive capacity. The chromosomes analyzed are those most likely to be altered. So we have some specific chromosomes that where there's any mutation or there's any alteration, either deletion, addition, or there is any genetic mutation in those chromosomes. One, the effect is very detrimental. So those chromosomes have been identified, and they are the ones that are prone to alteration. You have like chromosome 13, where it presents itself. You have chromosome 18, you have Patau syndrome, you have chromosome 21, which is Down syndrome. Then is also done on chromosome X and chromosome Y, which are the sex chromosomes. And although in cases of recurrent miscarriages, chromosomes 16 and 22 are also analyzed, where you have cases of recurrent miscarriages, because from some research, it has been discovered that chromosome 16 and chromosome 22, they contribute in recurrent miscarriages. However, after analysis, this spermatozoa cannot be used in analysis or in assisted reproduction to achieve pregnancy. So it is not possible to select or separate viable spermatozoa from those with chromosomal alterations. So this technique is just to know whether there is any genetic defect or there is any alteration or mutation in any of these chromosomes that have high tendencies of being altered. Then lastly, we'll talk about the pre-implantation genetic testing for aneuploidy, pre-implantation genetic testing for monogenic diseases, and pre-implantation genetic testing for structural rearrangement. All these are techniques that help in identifying if there is any genetic mutation. For these techniques is mostly done on embryos, either done on the level, if to reduce high level of mosaicism, it is advised that it is done on day 5 embryos, which are blastocysts. But however, the 3 cleavage embryos are also analyzed, and if there's any genetic mutation, it can be seen, whether it's chromosomal, which is done by PGTA, or it's gene mutation, single gene mutation, let's take note, which is done by PGTM. Some of those single gene mutation, like sickle cell anemia, hemophilia, albinism, they can be analyzed using PGTM. And where you have a translocation, or there's a mutation as a result of translocation, PGTA using structural rearrangement can be done. And such mutations or such translocations can be identified in the cells. So, but that for PGTA, PGTM, and PGTSR is done, like I said, the embryos must be generated. That means it's through an ICSI procedure. The embryos are biopsied, genetic analysis is carried out, evaluation of result is done by a geneticist, and appropriate counseling of the client before embryos are transferred. For recommendations, it is very important that the principles that led to prepare these recommendations reside in fundamental aims that professionals in IVF field should always bear these conditions in mind. One is that the need for careful diagnosis of infertility status, and secondly, the evaluation of risks for the child to be born after assisted reproductive technology and appropriate management of the pregnancy. And thirdly, consider all possible genetic causes of reproductive disorders. It is very important to consider genetic causes and the possibilities. And fourthly, encourage and support research works and grants. Then lastly, genetic counseling should be offered to all couples undergoing assisted reproductive technology. And then where it's needed, is also recommend that clinics should, as much as possible, assess or carry out pre-implantation genetic tests for clients that may be or clients that are suspected to present or to have genetic abnormalities. So in conclusion, many men and women affected by infertility or genetic disorders, genetic infertility that lead to decreased fertility have turned to ART, and recent estimates show nearly 5 million newborns have been assisted through IVF treatment. It is known that a woman's fecundity decreases with advanced age. Also, the modern tendencies to delay childbirth contributes to the increased use of assisted reproductive technology. Yet, there are several concerns about the safety and possible negative outcomes of ICSI treatment, such as pregnancy complications or terminations and miscarriages. So risk of various birth defects also. So and childhood developmental and mental defects have also been noticed. So it is important that all couples going through infertility, going for fertility treatment should be informed of the risk, the benefit and possible outcomes through the use of genetic testing and counseling. So when clients come to the clinic, it is imperative that proper counseling is done. Counseling may be offered whenever a genetic abnormality is suspected in either the male or the female partner, and should be provided whenever a genetic abnormality is detected. As more genes are being discovered, like I said, the genome project is complete already. However, the contribution of genes, because there are several genes that also contribute to some of these genetic disorders, the etiology of infertility disorders becomes better understood with research that is ongoing, and the management and treatment of infertility will improve as well. These are my references. And I want to thank my senior colleagues. I want to thank Akemini for her support. My MD, Dr. Akinsho, Professor Christian Makwe, Dr. Larry Waju, Uployed Medicals, Prof. Consults, Tosin Dolapo, members of Embryologist Roundtable, M2 staff members, my mentees, Victoria, Tolu, Ben, Stephen, Deme, Uche, Miracle, thank you for your support. Thank you for your contribution. And also thank the family. God bless. Thank you very much for this opportunity. Thank you very much, AFRH. And with this, I will come to the end of the presentation on genetic male and female infertility. I will hand over to Dr. Ikwani and Dr. Akinsoya for. Thank you very much, everyone.
Thank you so much, Mr. Joseph Olayinkaiwa, for giving that lucid presentation at the microscopic and cellular level. I must commend you for the thorough work done. So I'll hand over to my colleague, Dr. Femi Akisanya, to lead the next section of question and answers and then to conclude.
Once again, I join the members of AFRH to thank Mr. Joseph Uwa for these lucid, eloquent, and very rich presentation. It's really been very deep. And I want to believe that everyone that listened to the presentation have accumulated a lot of knowledge. Well, looking at the question and answer area, there isn't any question in the chat yet. I want to believe that the presentation was indeed lucid and everyone understood. But if there are still questions in areas that are yet to be understood or that need to be clarified, I invite everyone to please put in question in the chat so that we can pick it up. Well, in the absence of any question, I will turn it over to Dr. Ikwani to carry on.
Okay, I think somebody asked the question.
Oh, okay. 1 second. I think I'm seeing one now. Okay. I have one question here now. It says, in the case of Turner syndrome, what embryo stage and grade are most suitable for accurate PGT testing? And a second one says, okay, yeah, for a man with Klinefelter syndrome undergoing ART and planning PGT to prevent passing on the condition, is day 3 or day 5 embryo biopsy recommended?
Okay, thank you very much. So the first question is in the case of Turner syndrome, what embryo stage and grade are most suitable for accurate PGT testing? Now, earlier I said that for clients that present with Turner syndrome, the chance of them using their egg is very low. And so in case where they generate embryos, the 5 embryos are most, or okay, just a moment. So the 5 embryos are advisable. One, because the level of mosaicism, like I explained, is reduced in day 5 compared to day 1 and day 3 embryos. So day 5 blastocysts are recommended. For a client also that is presenting with Klinefelter syndrome, it is also advisable or recommended that day 5 embryos fully expanded blastocysts. One is that the level of differentiation is higher with day 5 compared to day 3 embryo. So where embryo transfer will be done, you are also assured that you are transferring the best quality embryos to improve the chance of implantation. So for both cases, day 5 embryo biopsy is recommended. Thank you.
All right, Dr. John Ikwani.
All All right, thank you so much. But it's quite engaging. Though we would have loved more questions to enrich this session, but I think the presentation was quite engaging and detailed enough for us to engage. So thank you very much. I think at this point, we're going to bring it to a successful conclusion. I think the Secretariat is going to share a list of our social media handles, and we're all encouraged to follow on Instagram, YouTube. I think it's here on the screen. Please, we encourage all members of AFRH, home and abroad, to follow us on our social media handles to be able to keep abreast of our activities and upcoming events. I thank you all on behalf of the Secretariat, Central Executives of the AFRH, for attending this rich webinar. And we'll be glad to have you again in the next 2 weeks for the next session on the genetic series of this presentation, which we're hoping will be compiled up later by the editorial board for our AFRH fertility textbook in future. So thank you very much for attending, and we look forward to seeing you next time. So have a great day, and you're welcome. Thank you.
Thank you very much. Thank you very much, Dr. Femi Akinsanya. Thank you, Dr. John.